Hello, my name is Rana, and today I will be talking about the topic of inherited diseases. So, deoxyribonucleic acid, otherwise known as DNA, contains the genetic and biological information that makes each species unique and is made of chemical building blocks called nucleotides. These nucleotides are made of three parts: a phosphate group, a sugar group, and one of four nitrogen bases. These nitrogen bases are found in the nucleotides or adenine, thymine, guanine, and cytosine, and the formation or sequence of these bases will determine the biological instructions that are contained in a strand of DNA. So, as an introduction to protein synthesis is the fundamental two-stage biological process that uses genetic instructions from the DNA to assemble amino acids into polypeptide chains. Protein synthesis involves transcription and translation in order to allow cells to build proteins which are needed for function, structure, and regulation. Transcription takes place on the nucleus where the DNA is unzipped and a specific segment of DNA, otherwise known as a gene, is copied into a complementary messenger RNA, otherwise known as mRNA, molecule by the enzymes RNA polymerase. Translation is where mRNA is decoded in a ribosome to produce a specific amino acid chain. This process converts genetic code into functional proteins through three different stages: initiation, elongation, and termination. The role of DNA in protein synthesis is to provide genes which will dictate a specific amino acid sequence in the process of protein synthesis. This is then transcribed into messenger RNA, which acts as the essential medium ground through carrying genetic instructions from the DNA within the cell's nucleus to the ribosomes in the cytoplasm. Transfer mRNA is an adapter molecule composed of mRNA, which serves as the physical link between mRNA and the amino acid sequences of proteins. The way tRNA does this is through carrying an amino acid to the protein synthesis machinery of the cell, otherwise known as the ribosome, as directed by the codon on mRNA. Ribosomes then translate the mRNA into a specific sequence of amino acids and then form polypeptide chains that fold into functional proteins. Gene expression is the process by which the information encoded on a gene is turned into a function. For example, a DNA strand may look like this. It may have a promoter and operator, some genes, and then a silencer. The promoter is a region of DNA that initiates the transcription of a certain gene. For this transcription to occur, RNA polymerase must attach to the DNA near the gene. This is because promoters contain a specific DNA sequence that provide a secure binding site for RNA polymerase, for proteins called transcription factors. The operator acts as a molecular switch, allowing or preventing RNA polymerase from describing genes, and the silencer induces or shuts down the transcription of specific genes by binding to repressor proteins, acting as a stop switch. Transcription factors are proteins that bind to DNA sequences to modulate transcription of target genes. This means that they can control the rate of transcription of the genetic information from the DNA to the mRNA through binding to a specific DNA sequence. Their function is to regulate genes and to make sure the correct genes are expressed at the right cell, in the right timing, and in the right amount. A repressor is a DNA binding protein that inhibits the expression of one or more genes through binding to an operator. A DNA repressor blocks the attachment of RNA polymerase to the promoter, which therefore permits the transcription of genes into mRNA. A point mutation occurs in a genome when a single base pair is added, deleted, or changed. While most point mutations are benign, they can also have various functional consequences, including changes in gene expression or alterations in encoded proteins. Point mutations can be caused by DNA replication errors or mutagens within DNA replication errors. DNA polymerase can insert the wrong nucleotide or fail to correct mismatches. With mutagens, certain chemicals can modify DNA bases or interfere with DNA replication, such as UV radiation or smoking. Additionally, chromosomal alterations can occur, which is a larger scale mutation that only happens within cell division. So after a point mutation, there are four different potential outcomes: a silent mutation, a missense mutation, frameshift mutations, and nonsense mutations. Each of these outcomes will have different effects. So with silent mutations, they will change a singular nucleotide base, causing a change in the DNA sequence, but the codon will still code for the same amino acid. meaning that the protein will have the same structure and the same function. Positive about this is that they contribute to a significant amount of genetic diversity within a population without causing harmful changes to the phenotypes. Negative about this is that they can affect the speed of protein synthesis due to the replacement of the codon. Slowing of protein synthesis in the ribosomes can then lead to a pausing of the protein synthesis, which can lead to the proteins being incorrectly folded. So within missense point mutations, they occur when a nucleotide substitution changes an amino acid. Missense mutations have variable effects but can lead to a decreased or altered protein function. Positive about this would be the rare and unique beneficial aspects that come with this mutation, which can enhance an organism's survival and adaptability. But a negative would be that how they can disrupt the protein structures and functions, which can lead to genetic diseases and serious health conditions, like sickle cell anemia, cystic fibrosis, muscle wastage disease, and neurological diseases. Insertions and deletions result in frameshift mutations, meaning that the codons are read at the ribosome. The codon reading frame is shifted by one codon, resulting in a completely different amino acid coded for from this mutation onward. The resulting protein will be significantly altered and usually non-functional. Positive about this frameshift mutation would be the rare case of disease resistance, and through a mutation in a specific gene can create resistance for a specific disease. Through using HIV as an example, due to frameshift mutations in the chemokine receptor gene CCR5, this creates a non-functional entry receptor for the virus, therefore increasing the person's resistance. A negative about frameshift mutations would be the potential increased risk of cancer, as an effective frameshift mutation if they encoded in tumor suppressor genes. If a frameshift mutation happened in there, it can promote an uncontrolled cell growth, leading to cancer. For nonsense point mutations, they occur in DNA when a sequence change gives rise to a stop codon rather than coding for a specific amino acid. Positive about this is that theoretically a nonsense mutation could shut down a harmful protein, such as a disease influences protein or a gene that produces a toxin. Negative about this is that they produce the premature stop codon, which can be detrimental to the protein synthesis process, resulting in unfinished or unfolded proteins which have limited or no function at all. Mendelian inheritance refers to the biological principle founded and published by Gregor Mendel in 1866 based upon his findings through experiments with pea plants. Mendelian genetics describes the way the traits are passed through generation from parents to offspring through alleles following predictable patterns. Specifically, these predictable patterns refer to the laws of independent assortment, the laws of dominance, and the laws of segregation. The law of independent assortment refers to how genes for different traits are inherited independently of one another, meaning that the inheritance of one trait, such as height, isn't influenced by another trait, such as skin color. The law of segregation refers to how each organism carries two alleles for the trait, which separate during the formation of sex cells, otherwise known as gametes. As a result, each gamete carries only one allele, and fertilization will restore the two-allele pairing in the offspring. The law of dominance refers to how, in a heterozygote having two different alleles, one allele can hide the presence of another, meaning that the dominant allele is expressed while the recessives are hidden unless the organism is a homozygous recessive. These are inherited when one or both parents give mutated genes to the child through inheritance, but there's different patterns as to how this can happen. For example, in autosomal dominant only, one parent needs to pass a faulty gene for the child to have a disease, or in an autosomal recessive, both parents had to pass the faulty gene for the child to develop a disease. Another way diseases can be inherited is through the X-linked inheritance, which are mutations that only occur on the X chromosome. These diseases are more common in males due to how they only have one X chromosome, as they are XY, which compared to females who have two chromosomes, XX. This means that females would need the same faulty gene on both X chromosomes for there to only be a substantial effect. But because males only have one X chromosome, it only takes a faulty gene on the X chromosome to affect them. Phenylketonuria is a rare but treatable inherited metabolic disorder that prevents the normal breakdown of protein. PKU is an autosomal recessive disorder, meaning that it's inherited by both parents having copies of the faulty gene for PKU, and for one from each parent being passed on to the child. PKU isn't a sex-linked disease, meaning that both parents must have a mutated copy of the gene for this condition to manifest. Gender is determined through the 23rd pair of chromosomes, which are the sex chromosomes. Throughout the process of fertilization, the male sperm will determine the sex of the child because males carry both an X and Y chromosome and has an equal chance of passing either on. Females only have an X chromosome, meaning that the rate of the fertilized eggs being male or female is a 1-to-1 ratio, otherwise known as 50-50. The XY and XX chromosome pairings are standard. However, it's important to note there are certain exceptions where variations can occur, such as Turner syndrome, where a female only has one X chromosome, Klinefelter syndrome, where a male has an extra chromosome, XXY, just as an example. Sex-linked diseases are inherited through mutations on the sex chromosomes. For example, hemophilia is a recessive sex-linked disorder that's caused by a mutation on the clotting gene on the X chromosome. Due to this disease being a mutation on the X chromosome, males are more likely at risk due to having only one X chromosome. In terms of X-linked diseases, males are more at risk of these disorders due to having only one X chromosome, meaning that unlike the females who have two copies of the X chromosome, the males still have to rely on the singular X even if it has faulty genes. Chromosomal abnormalities are genetic disorders caused by extra or missing portions of DNA that affects the 46 chromosomal structure. For example, in a trisomy, there's an extra chromosome out of a pair making a triple, which can cause genetic disorders such as Down syndrome, Edwards syndrome. Alternatively, monosomy is when there's only one chromosome present in instead of a pair and can lead to a disorder such as Turner syndrome. Genetic screening is done through the analysis of a biological sample such as saliva, blood, or cheek cells, and is done to examine a person's DNA proteins or chromosomes for possible mutations or variations that could be linked to diseases. All of the analysis will take place in a lab where the DNA will be isolated and various methods may be used to test the sample. For example, a molecular test will look at the small DNA mutations or analyze large parts of the genome in a chromosomal test. The structure and number of chromosomes will be checked and analyzed for any abnormalities. DNA recombinant technology works by isolating a specific gene and inputting it into another organism, such as bacteria, to create an organism that can create a larger volume of that specific gene. Through using this technology, it can treat certain medical conditions through creating human identical substances such as hormones, antibodies, and cloning factors. As an example, putting a human insulin gene into a bacteria can promote faster growth, which when compared to getting insulin from pigs or cows, the bacteria can grow at a faster rate and be given to type 1 diabetics to help them manage their medical condition. DNA recombinant technology has raised some concerns around ethicality and morality due to multiple factors such as altering an organism's original genetic code, accessibility costs, and potential risk factors. For example, a prominent moral issue is consent and consensual modification of human genes and the risk of exploitation. This is argued due to a single modification potentially affecting a whole genetic timeline, which causes the question of this technology becoming a consumer product where parents can alter the babies through changing the eugenetics. This is seen as another prominent moral and ethical issue, especially with the regulation and how far this technology should be regulated. Alternatively, another moral and ethical factor about DNA recombinant technology is the cost. The gene therapy has a list price of $1.68 million per patient, which is subject to the kind of treatment and how long it's needed for, but still raises important questions if the taxpayer should pay for these kind of therapies or the individual person, which leads to more moral and ethical issues about affordability, accessibility, and the general welfare of the people needing to pay for these therapies. The use of human embryos in research, especially when creating or destroying them, is subject to intense religious and moral obligations from multiple different backgrounds due to its individual people's beliefs. But in terms of research, it's arguable that DNA recombinant technology wouldn't be as advanced if human embryos weren't included in the development and research of this technology. The moral issue around human embryos depends if they are being emphasized in a person's mind as being a biological human or having a consciousness. As a last ethical issue, having genetically modified food or GMOs released into the wild environment could have significant effects on the ecosystem, as well as the agricultural system, such as disrupting the natural ecosystem, having unpredictable ecological consequences, and potentially affecting people's income, such as farmers and those who work in the agricultural sector. While having GMOs have been beneficial in plants growth resistance, harsh weather, increase in the plant's yield of crops. It also has greater risks alongside that, such as altering the genetic code of the plant. It could also help make them more resistant, but it could also somehow affect the wider range of organisms that rely on that plant.